Sir, shall we start, sir? Basavegowda, sir. Sir, shall we start, sir?
Okay, no problem.
Okay, sir. Thank you, sir. Very good morning to all, and I welcome you all to the 6th day of 10 days national level online training program on emerging trends in seed production technology and quality control framework for effective seed supply chain of horticulture crops. Today we have two lectures in the scheduled on this day. That is, the first lecture will be on post-harvest seed handling and seed processing of horticulture crops, which will be delivered by Dr. Basavegowda, Special Officer Seeds, University of Agricultural Sciences, Raichur. Then followed by, we have another lecture on perspectives and problems of synthetic seed production in horticulture crops, which will be delivered by Dr. Shivayogi Ravlad, Assistant Professor of Seed Science and Technology, University of Horticultural Sciences, Bagalkot. Now, I am welcoming all you to the this session. Now, with us we have very eminent person who is practically involved in seed processing aspects. That is Dr. Basavegowda, sir, who is a man with practicality in seed processing, because sir is instrumental in establishment of most of the seed processing plants under University of Agricultural Sciences, Raichur. Sir, on behalf of University of Horticultural Sciences, Bagalkot and College of Horticulture, Bidar, I am welcoming you to this session, sir. May I now invite Dr. Satyanarayana, sir, Assistant Professor of Entomology, to introduce Dr. Basavegowda to participants. Over to you, sir. Satyanarayana, sir, please.
Good morning to everyone and all. So first of all, I would like to make a mention, before I could actually start reading the pre-biodata of Dr. Basavegowda, sir, I would like to make a mention that Dr. Basavegowda, sir, has been my teacher when I was doing my BSc in agriculture at the College of Agriculture, Raichur. So which was an affiliate of the University of Agricultural Sciences, Dharwad then, but now it's a constituent college and the main campus of the University of Agricultural Sciences, Raichur. So Dr. Basavegowda, sir, did his PhD in seed science and technology, and presently is working as a special officer and university head for the seed unit at UAS Raichur, and he has 31 long years of experience in seed production, research, teaching, and administration. He is the founder for the establishment of independent seed unit in UAS Raichur, and he has been instrumental in developing a unique seed production and distribution system in the university. And to his credit, Dr. Basavegowda, sir, he has established advanced seed quality analysis and research laboratory and 11 seed processing plants with the large-scale seed storage facilities at different research stations. And Dr. Basavegowda, sir, has served as a principal investigator for the many outside funded projects to the tune of 16 crore rupees. And sir has guided more than 20 the postgraduate students at the MSc level and 3 the PhD students as well in the seed science and technology. And Dr. Basavegowda, sir, has published more than 120 research articles which are of international repute, and he has published 16 books and many technical bulletins as well. And Dr. Basavegowda, sir, has delivered 34 radio talks and 45 TV programs. So, and apart from having done all this work, and Dr. Basavegowda, sir, has visited several countries world over, and to mention few among them is the Germany. The second one is the Switzerland, and he has also traveled to the countries like the Sri Lanka, Thailand, for an invitee to study the seed systems and present his research papers in many international conferences. And Dr. Basavegowda, sir, has several recognitions to his credit. And the first and foremost, he has been the recipient of the Fellow of Indian Society of Seed Technology and Incentive Award for the US Raichur, Best Performance Center Award from ICAR, IISS, and Best Extension Scientist Award from the US Bangalore Alumni Association, and Lifetime Achievement Award from Society of Tropical Agriculture, Review Excellence Award from the ARCC, Eminent Scientist Award from the SVWS, Rashtriya Gaurav Award from the IFS, New Delhi, Distinguished Scientist Award from the BIF as well. And Dr. Basavegowda, sir, has developed the seed unit at UAS Raichur as a model green unit and organized many training and workshops for the benefit of the seed growers, producers and officials involved and developed 18 technologies which are useful for enhancing the seed quality and productivity. So this is the brief biodata of Dr. Basavegowda sir, and now I would request Dr. Basavegowda sir to take the charge for the further proceedings. Sir, please.
Okay, thank you for nice introduction actually. Before starting my lecture, I wish you all the participants wish you happy new year, and also I am thankful to all the, whether it is audible, visible?
Sir, your
Hello?
screen is not visible, sir.
Not visible, huh?
No, sir. Kindly share your screen, sir. Share
Just a minute. Where is it? Visible ಆಗ್ತಾ ಅಂತ. Is it visible?
Yes, sir. Now it is visible, sir.
Okay. I thank you very much for the nice introduction from the, and also Dean College of Horticulture Bidar is my classmate also, and all the officers including Honorable Vice Chancellor UHS Bagalkot for providing opportunity to share my some information about the post-harvest handling of seeds. Hello?
Sir.
Hello?
Sir, it's audible, sir. Go ahead, sir, please.
Audible, okay.
Yes, sir. Yes, sir. Please go ahead, sir.
Before starting the lecture, actually, for the benefit of outside Karnataka, I want to tell about the University of Agriculture Sciences at Raichur. Okay, US Raichur, it is one of the third agriculture university established in northeastern part of Karnataka. Now it is called as Kalyana Karnataka, consisting of actually 6 districts actually. Raichur, Kalburgi, Yadgir, Bidar, Koppal and Ballari. Now one more district is divided from Ballari, that is Vijayanagar district also. And that is one of the, for slogan, greenness is Asire Usuri is the slogan of the University of Agriculture. Our university consists of 4 colleges. One at Raichur, Bhimrayangudi, Kalburgi. Recently in Gangavati also one college in Koppal district started one. And 1 agriculture engineering college at Raichur, 13 research stations, 7 Krushi Vignana Kendras, and 6 agriculture extension centers. After the formation of the university developed many varieties in pulses, mainly in pigeon pea, chickpea, paddy, and all actually. Among the different varieties, actually our pigeon pea, that is a wilt resistant TS3R, it covered more than 70% of the area in Karnataka actually. Then our green gram VGS9, that is also one of the 60 to 70% area is covered by green gram varieties. Then our paddy Gangavati Sona, that is one of the leading varieties covered actually, and other varieties also covered after the developed by the university. This is of the view of the university where actually Karnataka, it is the northeastern part of the Karnataka. We can see actually this is the area where earlier it is called as Hyderabad Karnataka. Now it is a Kalyana Karnataka area where our university is working. Then about the university actually now, whether it is audible? Hello?
Yes, sir. Yes, sir. Clearly audible. Please go ahead, sir.
Okay.
It's audible.
Yes, clearly audible, sir.
Okay, thank you. Then about our unit actually, after the bifurcation from the US Dharwad, we started the independent unit actually system actually. Our model is actually decentralized system actually. Normally in university, normally will concentrate in one center actually. You want decentralized in all the 6 districts. Only breeder and foundations will be concentrated in the main centers. Others have our 6 districts or 11 units. We made in a different research station to decentralize and supply the quality seed to the all the areas in all the research stations and KVKs and all actually. And one more thing, after the university, we created one more VIGOUR program, that is a Kharif and Rabi seed day on May 25th and September 25th. Even other universities and also they will celebrate as a seed days. But we started in a particular day, that is May 25th for the Kharif season and September 25th. This is the idea is actually in order to provide the technical information before the sowing to the farmer. This is one of the unique program that is well accepted by the farmers of this region. And also we are operating like other university or NSP is operated in actually ICR seed product, seed project pulses, and also soybean also recently sanctioned by actually. The 17-18 our US Raichur also is awarded as the best performance centers actually. Then regarding the topic, actually everybody almost all are experienced and faculty members I've seen in the participants actually. Quality seed is actually win. Hello. Hello. Hello.
Hello, sir. Hello, sir.
Okay. Quality seed is one of the important component in agriculture.
The
actually.
slide is not visible. No?
Not visible.
But it is not properly.
What happened?
Sir, now it is okay, sir. Go ahead, sir.
Now it is okay. Now okay.
Okay, sir. Please go ahead, sir.
As you know, actually, that quality seed is one of the important component in agriculture. Actually, even actually with along with other improved technology, we can say actually as per the research and actually by use of quality seed itself, 15 to 20% yield can be that everybody know actually. Along with other technology, even up to 40% of the yield can be improved or enhanced by use of the quality seed. Then benefit actually everybody is actually knowing this is one of the important component in agriculture, horticulture crops, quality seed. We take about the providing the quality seeds to the farmer. Thereby we can enhance the farm income. Now government also planning for doubling the income. This is one of the component where we can include in the doubling the farm income use of the quality seed or seedling planting materials. This I can actually not much this one. Then we can know actually quality seed actually after release of the variety by the particular breeder and all actually that further maintenance and further multiplication is more important actually. Thereby during the lot of factors during the production, genetic, environmental, physiological and biochemical and other factors will influence the since it is a biological material, lot of factors than the genetic factors will influence the quality that has to be taken care while producing the quality seed. These are the different, this one may be only for the information. Then actually what are different practices actually? Then actually first one is maintenance breeding. Normally you can maintenance of genetic purity. Normally in a public system actually that area is most neglected, maintenance breeding. Only after release of the variety breeders and others they will concentrate for release. But that is a maintenance breeding is more important because without maintaining the genetic purity of a particular hybrid or variety, it is difficult to give a quality of the hybrid where you're getting the 100% genetic purity. That is more important. That has to be given more importance to promote the particular product. And also growing conditions where the varieties or hybrid is adapted. That is a selection of a good growing conditions and time and method of harvesting, processing and other operations also more important to supply the quality seed. These are the pictorial representation how quality is influenced by different factors. Actually, genetically, cultivar purity, longevity, and actually physical quality. Actually these are the factors. Physiological quality, pathological. This is also now as per the new Seed Act. That seed health is given more importance. That has to be given actually even for export of seeds and all actually. The seed health, especially seed-borne diseases and insect and designated, that is more has to be given more importance. That is why that you have to give more importance for the seed health also. Now actually quality of post. Now topic of this one post harvest handling actually. Now majority of we think that the production is more important. No, we want to supply the 100% genetically pure and highest quality seed compared to other private organizations. We should give more emphasis on the post-harvest technology. Post-harvest technology start from the harvesting till you supply the seed to the farmer. See, as per the data I have collected actually in India actually around 926 billion rupees US, that is 14.3 billion US dollars on account of post-harvest losses in India. Actually, that is one of the daily 19.4 million US dollar is paid in India. That is actually how the post-harvest is more important than the production also. Now actually, if you stop the production, not matter, but if you have to take the more care, if you visit any that public sector godowns and all, we can see how post harvest losses are going without taking any proper scientific method to conserve the materials. These are the important. This one we can see actually see different percentage of losses during the cereals 3.9 to 6%, pulses, fruits and actually where it is more important fruits and vegetables. This may be data for the commercial product. That is why even for the seeds also it is important to give more important for the post harvest handling. These are the different factors, actually what are the factors affecting the forest. That is most one is actually lack of harvest infrastructure and also limited knowledge on the good agriculture practices that has to be handled, and imperfect market knowledge and market and also inadequate market data. These are the important factors which influence the post harvest losses. This is also pictorial representation of post harvest losses in India. Actually, this is one of the area we have to give more importance. This is only, I told actually, this is only theoretical. You can actually, that is more important. You have to give the more important for the post harvest seed. See, different steps you have to take care of actually post harvest, starting from harvesting, dressing or extraction in case of horticulture crop, drying, grading, upgrading, seed sampling, also sampling, seed quality analysis, seed testing method, packaging, and so these are the different stages or steps you have to take care more importance in the while handling the quality of the material in during post harvest stages. See, harvesting normally will actually that you have to harvest more mainly on the physiological maturity where the crop is attained physiological maturity. The major problem in actual physiological maturity is nothing but the stage where you're getting the maximum dry weight. Practically, it is very difficult to harvest in horticulture and this one actually because of the high moisture content at physiological maturity. Then we can say as a the crop has to be harvested at harvest maturity where we can reduce the moisture content at field level. But if you have a facilities for drying and all, you should harvest the crop at physiological maturity. That is why I told you physiological maturity where the maximum dry weight actually, where you can get the maximum quality also and also in vigor. See, now actually physiological maturity, these are the indices different for the factors we have to see for the. Hello.
Sir, please go ahead, sir. Please go ahead.
Okay. These are the different indices where we can harvest the crop, seed crop for the, for example, tomato that turning of the skin to red or pink color, that depends on the variety and maturity stage. This, like that you have to see the different indices you have to follow where you can harvest the crop for the obtaining the highest quality. This has to be taken care. Even for all the crops, you should have a physiological indices where you can harvest the crop at physiological maturity. Then while harvesting also you should say actually it should be harvested and physiological, and also especially in horticulture crops it's better to harvest the crop at evening time actually to avoid the losses, and also crops should not be damaged while harvesting also, this should be taken care. See, some data we've seen actually how quality stage of the crop is influencing. For example, tomato we've taken different stages of harvesting. Some variety in L15 it is conducted in Raichur only. I've taken that is actually, you see, for example, different M1, M2, M3 actually. That fully matured actually that indicates you can see 100 seed weight and germination, even vigor and dry weight also it is more actually. That is why the stage of the crop and is more important in obtaining the quality of the material. The extraction methods also playing very important. Normally in horticulture crops, you are using the extraction method using the fermentation and all actually. That is the stage and concentration of acid you are using, and actually that is more important. These are the different methods actually. This method can be employed depending on the crops and all actually, different methods actually. The acid extraction method where normally hydrochloric acid is used actually for this one. These are the different, this one we can say here. See here actually that is different data is there actually. For example, for different crop that depends on the pulp. Tomato 25 ml per kg of pulp and 30 minutes and all. These you can standardized for your local season. This is only idea we can get actually how the concentration and also timings will influence the quality, and this you can determine and take care to obtain the quality of the material. These are the different steps actually. Then normally you should not use the metal container while going for the fermentation and all actually to affect the quality, and these are the stages we can actually. This is only alkali method actually. This also you can, but normally we'll use for the acid fermentation also. Here also they conducted studies actually. This is actually different studies actually. They found that 1% of HCl is compared to other methods. This is found actually. This you can standardize as per the data also. This is also different in eggplant brinjal. They conducted a method actually to studies actually. Here also you can see different fermentation methods how it will be actually. Even natural fermentation in case of that cucumber actually 94% germination is there actually. So you can see actually 94.5% in case of normal compared to other methods actually. That is why that method you selected for the different fermentation method is more important actually. That based on the crop you have to select the proper method of fermentation. The natural fermentation more approved than the other methods. Then drying also, this is more important in actually in case of any crop to maintain the quality actually. After drying actually, you can use the normal sun drying, and also now for the vegetables and all you can use the small dryers are available. This is more important to produce the different drying important discolor. Why drying is important after harvest actually, because of the high moisture content will affect the viability and discoloration, taxing, and especially seed health will be more affected if you don't dry the materials immediately after harvest to the proper dry content. This is also you have to take care. This is also you can, for example, if you want to dry the materials, if my seed moisture content is low, actually you have to use high moisture content in the bound form. High 43 so you can apply by as per the requirement. Here also different factors influencing the drying actually. This is actually depends. I already told initial moisture content, size of the capacity, and the depth of seed actually, seed rate, air flow rate, and air temperature and static. These all the different parameters. Normally, here actually, especially post harvest technology, in addition to the seed technologies are scientists, even post harvest engineers also they have to involve more in the maintaining these parameters in maintaining the quality of the materials. The method I already told, we can use the different drying methods and also artificial methods. Even now recently we can use the desiccants. Using this is more important especially for the horticulture crops. We can use the silica gel and all other the desiccants are available. This is one of the slow drying method, and also without damaging any seed material we can use mostly on the desiccants for the small packets and where we go storing the drying the germ blossoms. Here also moisture content, temperature, and all viability given. That is actually you have to restrict the temperature also 32, 43.3 like that actually, where you can maintain the quality during drying. Actually, this is different methods actually. Okay. Here also they given advantages and disadvantages different drying methods. Normally that you have to select based on the need, and also coastal area you can't try for the natural drying method actually, where you have to go for a artificial drying. There you have to select the different dryers and also temperature depending on the initial content of the moisture content and nature of seed also. See, this is now, for example, in Bidar also, that is where agriculture college is there. Now, for example, this is one of the model I can say actually that is established outside the university campus. Here also selection, this is showing actually how you can select the seed, equipment for seed processing actually. Here actually this chain normally in agriculture crops, we'll select a pre-cleaner, then a grader, then gravity separator, and now for soybean and all now they're taking the spiral graders, and also even horticulture crops also the chain you selecting for the processing, that is also more important actually. The chain, for example, there you can like the gravity separator and the color sortex also. Now majority of the that you can small seeded is there, you can hand clean it actually. Their color starter is also more important in horticulture crops, especially in case of hybrids. These are the different we can parameters we can consider while maintaining this one. Then while selecting processing equipment, that different parameters, maybe size, shapes, and density, and all. This is the principles you have to decide while selecting the equipment. For example, size, based on the size, you want to separate the seeds. This is mainly on the air screen seed cleaner. Shape also small graders were separate. That is a spiral graders also. So density you can use the specific gravity separate. Color now electronic colors are available. That is depending on your product actually, and also principal size actually you can select the or select the suitable processing equipment. These are the different parameters and all equipment you can select for the grading of the materials. Disc separators also can be. This is already depends on the broken seeds and the even gravity. I told this is mainly on the upgrading machines where after size grading, if any material same size using yes can seedling, this based on the density we can separate the seed material using the gravity separator. This is the model we can where it is like a normal based on the airflow rate, it will separate the material based on the density. Even this is one of the small equipment actually, spiral graders. We can separate round based on shape. This one actually, especially soybean, the gram, and also even bendy, and also they can separate it actually by farmer small seed. This is cost about 8 to 10,000 only per unit. It can be used for a small scale, this one for grading the material based on the roundness or shape of the material. Materials were exporting the seed materials, especially pulses and all, even all private companies they use even discolored and diseased seed materials can be separated based on the color background color you have to select there, that is electronic color surtex. This is one of the most useful equipment, especially for the hybrid seed separation or upgrading machine. This is one of the, I can say actually this is automatic seed treater, dryer and packing unit established unit actually, seed unit actually. Normally in a seed processing unit, especially in public sector, we'll use only mistomatic seed treater. While using the mistomatic seed treater, the seed treated material will be having high moisture content. Immediately you can't pack the materials actually. Then you are going for a manual drying and all actually. That actually you procure this equipment. This is actually, it will treat the material using the mistomatic. Then material will be moved through a dryer in a conveyor actually. After that, it will moving for a packing unit actually. The advantages is actually immediately after treatment, you can pack the materials without handling any manual handling. And also you can supply the material actually. Normally all the private sector you having this type of machine actually. This is for the public sectors also. We can upgrade the material. We can have efficiency in packing and very good this one system we can have a for upgrading the quality seed treatment and all. This is available in Raichur. This is one of the unit I can say in available in public sector in Raichur actually. We can pack around now agriculture crop over 100 tons we are packing per day actually. Even for horticulture crops also small this one actually. The advantages is immediately after treatment you can dry and pack the material. See, this is groundnut actually because this group is in horticulture crop. Because why I am showing actually, normally decortication system is having the single grading system. This is a double screen decorticator system. That is advantages is immediately after decortication, there won't be any hand cleaning of the materials because labor is very costly input now actually. Like that, you can equipment can be upgraded without having any manual. Here actually double screen means after decortication it will be graded actually. Directly you can supply the material without any hand grading also. Then after processing and all actually, normally people will say sampling. Sampling they normally say they will tell to processing plant in charge, they will tell the labor. No, it is one of the very scientific method we have to follow by itself. It is more important based on the sampling technique you are following, sampling equipment, different this one. Based on that, we can get the result. If you want to have a representative results, you have to have a correct sampling method and sampling layers also methods you are using. Here also different trays and the advantage is actually if you manually draw the sample, you may not reach the entire container actually to draw the representative samples. The best method as per my practical experience is actually to draw the sample. For example, 1 hour processing is there. Every 20-25 minutes you draw some samples like the 20, 25, 24 times you draw this sample, then you make it. That is one of the best method. Otherwise, you have to draw the samples using the sampling trayers and also intensity. That is more important. This is actually, actually how you can get the primary samples and composite samples and all actually. Based on that, this is more important. How you follow the sampling intensity actually. For example, up to 50 kg you have to have minimum primary samples. 51 to 500. This is as per the STAR rules. Normally, majority of the people were not following this one. This is also more important to have the representative result from the sample. This is also if you have a samples actually, based on that you have to draw the samples. This is also how you can working samples and all actually. My idea is actually that you have to, instead of telling to the other people actually to draw the sample, this is also more important. You have to supervise the sampling method based on the scientific sampling intensity recommended by STAR. See, now actually storage also now, this is one of the view we have taken from the private, this one actually on that. Use of pallets and stacking system also. Now actually wherever moisture content you have to use the pallets like a baby wooden pallet. Now because of the environmental concern actually you are not getting the wooden pallet. Now plastic pallets also, that is more important. Advantages is there actually, that migration of moisture content from lower way, and also aeration also. If you use the pallets actually instead of mat and all actually, the aeration from the ground it also will be actually. Here actually they given by step to handle by the mechanical handling. This is also more important. While in storage and all you have to give more important during the store, how to maintain the hygienic conditions and also scientific technique to maintain the quality. These are the genetic quality. Okay, this is okay. See, even next you are sending out samples actually. Here also actually, how you can going for the testing of the samples from the after post harvest actually. Normally testing method, selection of the testing method also, that you have to adopt as per the STA procedure. Whether it is a BP method, between paper method, top of the paper method, or this one actually, what is the sand method? That depends on the size and all you have to check the check method. And immediately after receiving the sample, normally you should give the decoding. Coding should be there actually. Coding then only, because the sample analyst from the lab actually should not know which sample from which producer the sample is given. That has to be coded and given to the lab. After this is the receipt of the sample, that has to be divided properly using the sample divider. Then you can go for the normal routine test is germination, physical purity and moisture. Now already told actually the seed health also is more important now for the export of the seed materials, and that is given more important. And also genetic purity by maybe using the markers and all you can go. Otherwise also growth test you can have a genetic purity. That is also one of the important. Why private people are more successful in seed industry? Because they give more importance on the routine test. They will have a want to have the objective to achieve the highest quality than the minimum required as per the seed act. Normally public sector will give only if it is minimum achieved, okay, it is passed in the, that may we can sell actually. But the private way they want to achieve 100% genetic and other quality parameters. That is why they are more successful. Here also in germination actually, here actually for example some people I can see actually they say germination means just viability actually, that is not a wrong. Here actually that is the ability to give the normal seedling, that is more important. That if you give the select the classification of normal and abnormal seedling during germination testing, that is also more important. That should be done by the technical expert. If you classify properly the normal and the abnormal seedling, then your failure performance of the seed material in a worst condition also, that this will be better. You won't get any complaint from the seed lot supplied. Here also paper and how you place the method. This is also wrong actually, where you have to zigzag position actually. That is a straight. You should not place the seed material. You have to provide the equal spacing also for the germination on the germination paper. The different method I already told, different paper. Even number of seeds you are placing for the germination. This one that also influence the seed material. For example, 200 seeds they told. 400 seeds replicate for each replication in a small seeded actually. For bold seeded, you have to use 50 seeds in 8 replication. That also will give a, for example, also actually, this is also classification of abnormal, fresh and odd seed. This is actually technique where you have to use based on the to get the accurate result. See, for example, this is one of the study we conducted at this one actually. We can see 25 seeds, 50 seeds, but for example, Kabuli. This is only example I taken. Bold seeds, if you use 100 seeds, even good seed lot, it won't give the germination. That is why selection of that is 25, 50 seed placing the, even though your lot is good, in while the wrong testing method, your lot can be failed actually. That is why selection of the proper method of seed testing and also number of seeds and also as per the ISTA, you have to follow scientific method to get the accurate result in the seed testing also. The moisture testing also, this is actually one of the method actually we can use. Normally you will use the digital moisture meter, but if you want to have a digital moisture meter is okay, that is an instant method, we can quickly you can get the material, but it is not accurate. You have to use the own method, that is a more accurate to get moisture meter. That is why hardware women depend on the temperature and all, you have to select the accurate method for testing the moisture content. And also one more thing actually, while they're sending the samples for moisture testing, you have to send one more sample along with the sample. One for the sample for the routine testing, germination, physical purity and all. Another in a moisture proof container for moisture testing. Otherwise seeds are hygroscopic. You will get the variation in the moisture content if you send the samples in a cloth bag or under for testing. Even quick viability testing also. Some of the time actually because of the dormancy and all actually you are unable to do the routine germination testing, then you can use the TZ test and all actually, where you can quickly you can declare the result based on the viability and all, you can market the seed material. This also you can know actually this is how it will be designated using the tetrazoline chloride test. Then now I told actually seed health is given more important, especially if the new seed law is implemented. There also you can use the dry examination of the seedling symptom, agar plate and blotter method. That is more important now. All our lab has to be updated for the seed health testing and also that we can supply the healthy materials and we can compete in the market. These are the different tests we can adopt for the evaluation of the seed health in the laboratory. Then VIGOUR, now actually that is a more important for the VIGOUR. Normally as per the Seed Act and all, for even VIGOUR for the, there is no standard for the VIGOUR. Still you want to perform in a, this one actually, for example, now revalidated seeds are there. Even though it will pass in the germination, routine germination testing, but if you supply and all actually it will fail in the field performance. To avoid that complaint and all, VIGOUR testing is more, this is only comparative test actually, that we can standardize and apply in the seed testing method so that we can compete in the market and also avoid the complaint from the farmers. Then seed evaluation and also lot of techniques are available now using the X-ray techniques and all actually evaluation based on the without non-destructive evaluation method for evaluation of the quality seeds, quality of the measure, for example, electromagnetic radiation. These different techniques are available, especially NMR and all that can be applied for the evaluation of the quality seed without any damaging that one, so that we can quickly, these are the electromagnetic radiations and different techniques are available. We can use for even for the detection of the seed health. This can be applied. Image, this one I can visible light and all this can be. Now this is the era where you can use the technologies for quick and accurate analysis of the seed quality. This also has seed quality and also the external features you can say that is already told. Even for the embryo, seed coat condition present now normally will conduct only routine. This is also well where actually you have to expert the science in this one to have a 100% genetic purity and so that will supply the quality material. The same thing you can apply for the different techniques also. Automated image capture and also you can have for the quality this one actually. Then X-ray technique you can use. It's also different views actually how you can see the seed, so that we can have a even seed health analysis. Other than quickly you can get the result based on the different techniques, so that normal, abnormal we can classify based on the embryo. This one you can image improvement actually. Pest and diseases also we can using the X-ray and then packing. Normally in a public sector, especially now horticulture crop, they are using actually. How you select the packing, that is more important in the seed industry. Normally packing materials are classified into pervious container, impervious and impervious for the vapor proof also. Now normally in agriculture crops and other bulky horticulture crops, you are using the cloth bag or cloth bag, that is a pervious container, where moisture content is more than 10 to 12% actually. That is for the short term. Especially hybrids and all, now for all private people are using the that high quality impervious materials actually. When you are going for the packing material with impervious content, the moisture content is more important actually. It should be 2 to 3% less than the recommended moisture. Then only we can maintain the quality. Otherwise, it is very dangerous. If you want to use the impervious material with the normal same moisture content, it will damage the seed material. That is why if you want to store the pack the materials in the impervious or vapor proof container, you have to take care about the moisture content. It should be 2 to 3% less than the moisture content of the previous container. These are the different types. Even in other this one now actually even heavy gauge paper bags are also available, where you can use the for packing materials. Impervious I told actually it is less than one, that is a polythene bag that can be used. If you want to store for a longer one, it should be more than 700 gauge it should be there, but you have to take care about the moisture content while packing the materials. And also recently in markets lot of bags are available, that is a Purdue improved crop storage bags, pro bags and all. Actually this is as a multi-layered bags, we can store the materials. You can actually here also the technique is same actually, where you are using the hermetic storage or impervious storage. You have to take care about the moisture content. Storage I already told actually, everybody is knowing actually, that is you have to use the Carrington thumb rule actually, where it should be 50-50, that is 50% RH, 50 degree Fahrenheit temperature. The sum of less than 100. That you have to take care about the storage actually, as by using the that Carrington thumb rule to apply for storage. Here also they given actually how moisture content during storage will influence the storability. That is when a very 1% reduction in moisture content will influence. That is why moisture also more important, that you have to take care in the storage. Now modified storage using the that calcium chloride up to 60% they throwing actually, even all you can see actually industry all chips and all they pack for a years together without any damage. This can be applied this technology for a storage of especially vegetables and other crops, where you can use the modified storage by controlling the carbon dioxide, higher concern carbon dioxide actually, even up to 60% they say that is you maintain the quality and actually. Then hermetic storage already told, that is a very you have to use, but only you have to care about the moisture content. Then also cold storages. Now cold storage is available in this one, especially Gubba cold storage in Hyderabad. They applied a lot of this one actually. These facilities can be used for the storage of normally. Here also the temperature is very big. There normally low temperature is better, but cost of the maintenance will be always more if you maintain the 5 to 6 degrees centigrade. Even for the liquid nitrogen also, that is actually for that is why cold storage establishing is okay, but maintenance also depending on the this one we can, for example, if you want to store material for only 1 to 2 years, that is only conditioned storage is enough. Conditioned storage means around 18 to 20 degree or 45 or to 45% RH. If you want to store for the longer period up to 5 years, actually 12 to 5 degree around 35% RH. Even if you want to for a longer storage, I told you minus 20 also you can use, or liquid nitrogen or minus 80 degree. That is for germplasm. That means your cold storage selection is actually depends on the your need. Actually, you want to store it for germplasm for longer period, you can establish for a 1 or 2 years. That is actually, but that is why the cost of maintenance depends. You have to take care while selecting the or establishing the cold storage also. See, this is one of the packing material I told actually, that is the controlled and all actually how packing material, impervious packing material. This is one of the study conducted in onion actually. That is actually, if you want to store in the impervious packing, definitely it will give the very good quality this one, but you have to take care about the, I already told, moisture content. It should be 2 to 3% less than the recommended moisture content. This is even the dehydrogenase activities also. That impervious container is maintaining higher enzyme activity. That is why VIGOUR will be more compared to the pervious container. Here also one study conducted actually, even that told, no, Raichur we identified one triple layered bag, I can see chickpea bag, seeds stored for 14 months without any damage. That is a triple layer bag. We are registering that one. That is also bag that is around only 50 rupees per quintal. That we want to promote at village level. Even that is more required actually. Now we are selling a seed with concept and all. Especially in case of varieties and all. Instead of farmers purchasing the seed for every year or every season from this one, if you train the farmers for production of the or selection of a varieties and at village level, and you can store by using this type of improved low cost storage bags, you can maintain the materials for longer period, so that our seed replacement rate also can increase, especially in case of high varieties. You can produce at a farm level and store and reuse without any reduction. This is also cold storage, I told. No, this is a commercial cold storage. Commercial cold storage, you mean that is normally established at 5 to 7 degree or 65 to 70% normally for storage of chili, commercial chili, tamarind and pulses. These are also studied actually. Even after 22 months, the seeds of chickpea is not affected. That means even commercial storage available in a this one, that also you can use for the storage of seeds. See, this is normal storage, that is commercial storage. You can see 22 months how the seeds are maintained quality without a big effect. You can see this is a normal condition where the seeds are infested and all the infested seed health and health of the condition and also you can see. This is also biotreatment, also castor oil they be treated actually. Even this can be used for horticulture crops also, some of the low cost technology. This is only 5 ml per kg. This is a biological method also we can use for the storage. That is a biological method, means without treating any chemical. One is use of impervious packing materials, then castor oil and all this can be. After that we can use as a edible purpose also. Castor oil won't affect the health also. Then you can see sweet flag also. Some study conducted, that also can be 10 to 20 gram per kg we can use for the treatment of seed. This is also one of the non-chemical method we can use for that. It's also same in this one. In this one. While concluding, actually, that means actually lot of technologies are available in the post service. This is one of the area normally public sector are neglecting post service technology, that we're giving more importance. It should be given more importance. And actually now actually second one is actually lot of technologies available in the industry, especially you have to go on the PPP model, public private partnership in adapting the technologies and all in packaging, seed coating and other technology. Thereby you have to give the more value to the our seed material, so that we will compete in the market itself. And also normally in public you will treat the materials, and if it is not sold, you cannot be consumed also. Non-chemical method also you can practice, so that that wastage can be adopted. Then I already told there is a scope to adopt the latest technologies available in the for quality evaluation, sampling and analysis and all. Then especially genetic purity testing actually normally it depends on the field, it will take a lot of time. Then molecular level and all that should be needed, and also then seed health also, quick analysis of the quality seed health testing also, that should be adopted so that our quality, normally we should target maximum quality than the minimum standard as per the seed rules and all. That is why, that is the difference between the public and private sector. Public sector, they will concentrate more on the maximum quality. Normally we want to meet the minimum quality. That your target should be to achieve the highest quality. And also as per the seed act and all, now lot of strict this one will be there actually. Regulation, maybe certification, now still it is under discussion, maybe compulsory, and also that is also we should ready. And also now all the varieties and all has to be compulsorily registered. That has to be, for that actually our public sector and private sector ready for the testing and compete in the market. Thank you one and all again. If any questions, I can ready actually. Thank you.
Thank you so much, sir, for your nice and very informative presentation on post-harvest seed handling and seed processing in general. With the permission of speaker, now the session is open for discussion. If any questions are there from participant side, means just unmute yourself and you can ask the questions.
Hello.
Hello.
Good morning, Basavegowda ji.
Good morning. Good morning.
It is VT Hegde and Happy New Year.
Thank you. Thank you.
Happy New Year to all, and I'm very excited, and it's a very wonderful talk. After a long time, I hear a complete story of the post-harvest and the importance and the management. It's a good presentation.
Thank you, sir.
Basavegowda ji, and also I appreciate all the good work you are doing in a Raichur unit, and I am a follower of you, and wonderful effort. That is the first thanks. And my question is, maybe many times you appreciated the contribution of private, and really it's a very good compliment for all of us. And as you mentioned, we are targeting for the highest quality. Minimum standard is one thing, and we always go for the best, highest quality. Maybe I am from a Nunams again. Maybe for the audience, we are selling the seeds by count.
Yes, sir.
You may be knowing, Basavegowda. And I have only two suggestions. Maybe now, as you mentioned, the seed health, and the seed health is one important factor where we have to consider. And you also emphasized the quality should start from the production. If the initial quality from the production is good, and it will help for the further easy for the post-harvest management, and also reduce the wastage. You rightly mentioned. And seed moisture and seed drying, maybe I would like to ask your help. Any solar drying project you are working out, because nowadays
post-harvest technology is having, sir. If you have any this one, you can mail it me also. We'll help you, sir. Our other unit is their post-harvest technology. They have developed the solar unit for drying.
Yeah, that is the need of the hour because of the fluctuation in the weather, and also the fumigation. Any recent advances in the fumigation?
Fumigation research you not done actually. That is actually there also not covered that actually. Now only you are using only regular sulfur. This one actually aluminum phosphide.
Yeah.
But repeated fumigation, definitely it will damage the material, sir.
Thank you. Good. Maybe sometimes I appreciate you to contribute and continue all the good work. Thank you, sir.
Okay. Thank you. Thank you.
Good morning, sir.
Good morning.
Happy New Year to all. I'm Siddharth Acharya.
Okay, sir.
Sir, I have a question.
Can the small and marginal farmers are encouraged enough for this mass and cash type of technology so that it can be easily available to them, so that they can have the better advantage of this new technology, sir?
For storage, no?
Yes, sir.
Even I told, no, we developed a small, simple, economical packing material. That is 50 rupees for 50 kg packing.
Okay, sir. Okay.
Without any carbon dioxide, normal condition only, they can store the material for 1 year.
Okay.
But only thing is actually, while storing the material in this one impervious bag, moisture content they should take care.
Okay, sir.
It should be properly cleaned and dried.
So farmers should be trained. And
if you want, actually, I can send the sample also. If you mail it me,
yes,
send the sample. You can try in your area also.
Okay, sir. Okay. Thank you, sir. Thank you, sir.
Okay, thank you.
Good morning and very good presentation. I am Dr. Chitra Pandey from Delhi, NPPGR. I am working as a
Tell me, madam.
vegetable crop curator.
Okay.
Sir, I would like to know, you have spoken about the image analysis.
Okay.
Okay, so that is recommended for which crop? Because there are so many things. So
That is
which
under
crop your people are doing?
No. That is only review this one. Actually, the technology is available. You have to standardize.
Okay, you are not practicing it.
Not practicing.
Okay.
That is not a practicing. That is
Okay.
available. We can practice also.
Yeah, it is available long back. It is there.
But still you are not practicing. Like our molecular work.
Yeah. In IRAC technology discipline, they are doing this work. And
but
sir,
still
one more
it is-
question.
okay, okay. Tell
I
me,
got
madam.
three questions. One more questions. Like 2 to 3% moisture means it is ultra drying. Okay. It is
No,
very
it is not
difficult.
ultra, not 2 to 3%. For example, for paddy, it is around 12% moisture content per the pervious bag. You have to reduce 3% from 12%. That means around 9%. Not 2 to 3% ultra drying.
Okay.
2 to 3% less than the moisture content recommended for the pervious container.
Okay. Yeah. Otherwise, it is wrong. No? It will take longer period if you try to reduce
2 to 3%
moisture.
less than the moisture content recommended for pervious container.
Recommended for pervious containers. Okay. Since I am working in NPPGR and we are doing long-term conservation, it is very difficult. 5%, 5 to 7% moisture content we bring,
That is only
and we have got very good drying facilities, and we are storing the seeds in the tri-layered aluminum foil packs. You have mentioned tri-layered packets for chickpeas. Some slide you had shown. So what is that one specification of that?
That is actually now, madam, that is under registration. We'll disclose it actually after registration.
Okay. Since
but other bags are available in the market, maybe fixed bag and grain pro bag that you can get in the market also. Similar to that, but 50% cost is less.
Okay, we are using for more than 4 lakhs germplasm lines. We are doing in hermetically sealed, tri-layered aluminum foil packs. So that was my curiosity to know. And it is a good presentation. Thank you.
Okay, thank you.
Hello.
Sir, good afternoon. Sir, myself Mahendra from... Hello.
Excuse me, sir.
Yes. Hello, sir.
Hello.
Sir, myself Mahendra from Maharashtra, Dr Basavegowda University.
Tell me.
Hello, sir.
Hello.
Sir, in one of the slides, you have mentioned that nowadays private companies are practicing X-ray technique for germination test.
Not X-ray for germination test. That is only damage analysis, seed health.
Yes, sir. So, sir, what is the dose, sir? Dose. I want to know the dose of that X-ray, sir.
No. That is actually research is there actually. You have to practice that one actually, but you have to compare with the other test also.
Okay.
Then only you can use method.
No, sir. In BRC, sir, they are doing the mutation and they are using the different doses. It is 100 grain, all that.
That is for the breeding technique.
Yeah.
See, for example, this is in medical field also. They will use the X-ray technique for the analyzing the human body and all.
Yes,
same this one there you are applying already research is this is the old research only. X-ray is not new one. If you pass it actually, what is the damage and also internal seed-borne pathogens also you can know it actually.
Okay, sir.
For that you can know actually this is for the comparative this one about your quality material quick method.
Yes,
without any damage.
Okay. Thank you, sir.
Okay.
Hello, sir. I am Rajani, sir, from Kolar, Karnataka.
Okay.
Sir, actually in our area it is a mango area, sir.
Okay.
Especially during the season, we need to sow more this one, sir. For root stock, we need to sow more number of stones, sir. So sometimes what happens, sir, due to labor problem, it may extend to 1 month, sir. So for that, if it is any treatment is advisable for extending the viability of the stones, sir, mango stones.
Variety mango, we don't have any idea actually.
Yes, sir.
You want to have a treat? How many months you want to extend?
Up to 1 month, sir.
Horticulture group only can actually. We're not having any data on that.
There is no problem with that for up to 1 month, sir. Raju sir. You can store in open condition and in the shade, and no problem. Most of the nurserymen they are practicing that method only. Sir. There is no such such methods to improve this viability of that mango fruits.
Okay, okay. So there is no such treatment, sir.
If you want to increase more than 1 month, up to 1 month, there is no problem, sir. Most of the nurserymen people, they are practicing that.
Actually, whenever we get labor problem, sir,
Of course, you mentioned 1 month. That's why, of course, that is up to 1 month. There is no problem.
Sometimes it is around 1 month, sir. It may be extended to more than 1 month. So, in that case, is there any
priming of that this stone priming?
Stone priming. Okay.
Potassium nitrate, potassium sonite, or some vitamins, or some growth regulators. Then you can. But after priming, immediately you have to go for planting.
Okay, okay.
Okay, thanks.
Okay, okay, thank you.
Hello.
Sir, Pedavalli Subbarao has raised his hand. Are you there, sir? Pedavalli.
Hello.
Hello.
Sir, this is Dr. Shivrama Krishnan from JNKVB Jabalpur, sir.
Tell me, sir.
Yes, sir. Very happy New Year, sir.
Same to you, sir. Same to you.
You might recall me, sir. You came to our Institute for Monitoring.
Yes, sir. Yes, sir.
In AICRP NSP program.
Thank you. Remembering now. How are you?
Sir. I am very glad to hear your talk, sir, and it is very informative for a new scientist like us.
Okay, thank you.
Okay, sir, my question is that, sir, in our area, the problem of post-harvest storability in case of soybean is there. So what kind of post-harvest intervention we can make in order to increase that storability, sir?
See, now you told actually soybean that post-harvest, first you have to start from the harvesting itself.
Yes, sir. Yes,
First one is, that is a very sensitive crop, actually delicate seed.
Yes, sir.
While harvesting also, harvest from the physiological maturity, then actually you are using combiner or manual harvesting?
Sir, we are using combiner, sir.
Combine, there are RPM also you have to control.
Yes, sir.
Normally we don't bother about the RPM.
Yes, 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 sir.
Jabalpur is done lot of work on the soybean only.
Yes, sir. Yes, sir.
There you have to RPM of the combiner also you have to regulate it.
Okay,
then handling, further handling.
Yes, sir.
Handling also. Then even for all operations, it should be handled like a baby.
Yes, 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 sir.
Already technology is available, only you have to adapt it, that's all.
Okay, okay, sir. Okay, sir.
And especially now major problem is you are getting the rain during harvesting. That is a major problem now. Everybody is facing now.
Major problem. Yes, sir.
And also processing also you should have a separate processing equipment for the soybean. Instead of that all this one spiral graders I told, no, that small scale you adopt that one actually. Even that elevators, bucket elevators you should not use. That is only zigzag elevators will be there.
Yes, sir.
That different techniques are available that you have to adopt starting from the harvesting to the final handling. Even stacking height also, processing, handling. That I told, no, it is like a baby, you have to handle it.
Yes, sir. Yes, sir.
That is an integrated practice you have to practice.
Okay. Sir, what kind of packing material we can use, sir, for soybean? That is
packing material, no, that all depends on your initial quality. If initial quality is damaged, even using by other good packing material, it won't improve the quality.
Yeah.
But definitely the super improved bags are there. No, they are performing well.
Yes, sir.
Polylined bags and all, even for the market also, their separate bags are there.
Yes, sir.
But only moisture content, and also before that, what is your in-cell quality?
Yes.
You can maintain the quality, you can't improve the quality in storage.
Yes. Maintain करेंगे. We can increase the storability only, sir.
That's only. But
Yes,
in-cell
yes.
quality is important. No, if you have 70% in the in-cell itself,
Yes,
that
yes,
cannot be
sir.
stored for further.
Yes, sir.
Okay,
sir. Okay, sir. Thank you, sir.
Okay, thank you.
Yes, sir.
Thank you so much, sir, for nice deliberation. With
Okay,
this, we
thank you.
are
Hello.
closing this
Hello.
presentation.
Hello.
Somebody is telling.
Hello. Good morning, sir.
Good morning.
This is Devanushi Datta from NSP Assam Agriculture University, sir.
Okay,
Very
tell
nice
me, madam.
presentation, sir.
Thank you, madam.
So I have a query, sir. So in one slide, you have shown the use of X-ray or gamma ray, that is the radiation technology, for the seed to see the internal seed microflora.
Internal damage and microflora and all.
Okay, sir. So what is the impact of seed germination or the viability of those technologies, sir?
No, that is actually to detect the internal damage by the seeds.
Okay, sir.
Okay, that technique you can use for the assessing the internal damage.
Okay, sir.
That is one of the technique for seed health only.
Okay.
Quick technique.
Okay, sir. That the X-ray, the machine, what is used for our human body, that same machine can be used or other?
But you can Google it, madam. That's actually we are not done actually. That is only based on review actually. But the same machine you can use, only wavelength will be different, I think.
Okay, sir. Or one more question, sir. For chickpea storage, you have used the triple layer bags, sir. So what may be the cost of those bags, sir?
But only 50 rupees for 50 kg packing.
50 rupees, 50 kg bag.
1 rupee per kg.
Oh, okay, sir. Okay, thank you very much.
Okay, thank you.
Sir, hello.
Hello.
Sir, whether use of sulfurs like fumigants, aluminum phosphide, will reduce the germinability of seeds?
No. Once you can practice, actually, there also that moisture content is more important. It's the moisture content of a seed. If you use the seed moisture content, higher moisture content, it will damage. And repeated fumigation definitely will damage the seed material, especially in oil seeds and all. One fumigation, not problem. 1 or 2.
In pulse crop, in pigeon.
1 or 2 you can use. 1 or 2 you can use.
Okay, sir. And in closed environment means inside the seed bin or in airtight condition, how many days it can be stored?
That storability depends on so that is all I told. No, in-cell quality of the material. Then if already material is infested, you can't store the materials. Then moisture content. If the material in-cell quality is good, moisture content you reduce to 2 to 3% less than the ambient this one. Definitely 2 years you can store in the airtight condition.
We have experienced that in where air movement is there in airtight container. In airtight container, germinability reduced faster. Why?
No. That is why I told actually. For the storage, if it is material should be clean, you should try it properly. 2 to 3% less than the moisture content recommended for pervious. Definitely 2 years you can store the material in the impervious container.
Maybe our moisture content may be more.
That is more important. Moisture content is more important.
Okay, sir. Thank you, sir. Thank you very much.
Thank you so much for nice presentation, sir.
Thank you.
With this, we are closing this lecture. May I now invite Dr. Surem sir to propose vote of thanks. Over to you, sir.
Thank you, Dhananjay. It is my privilege to deliver the vote of thanks to Dr. Basavegowda sir for nice delivering the lecture and with good presentation. So on behalf of University of Horticulture Science, Bawalkot and College of Horticulture, Bidar, I heartfelt thank to the Dr. Basavegowda for your nice presentation and good lecture, sir. Thank you, Honorable.
Thank you all.
Thank you,
again, I wish you all the happy new year.
Thank you, sir. Wish you the same, sir.
Thank you.
Thank you so much, Dr. Surem sir, for proposing vote of thanks. Now with us we have another speaker, that is Dr. Shivayogi Ravalad, Assistant Professor of Seed Science and Technology, University of Horticulture Sciences, Bagalkot, who is going to deliver his lecture on perspectives and problems of synthetic seed production in horticulture crops. Sir, on behalf of College of Horticulture Bidar and organizing team, we are welcoming you for delivering this lecture, sir. May I now invite Dr. Eknath Kamble, Assistant Librarian, to introduce Dr. Shivayogi sir to the participants. Over to you, sir.
Thank you, Dr. Dharan sir. To all, I introduce today's research specialty, Dr. Shivayogi Ravalad, who is Assistant Professor and Nodal Scientist, Seed Science Unit, UHS Bagalkot, since 10 years. Sir is graduated from